You know, it's a moment that every single pet owner absolutely dreads. You're just petting your dog or your cat, and your hand just stops. It feels something that wasn't there before, a lump, and your heart just sinks, right? Well, that's the start of a mystery. It's a case that really needs to be solved. So let's dive in and see exactly how veterinary detectives crack the case of the lump. So you're there, standing in the vet's office, and the big question hangs in the air. What is this thing? Is it benign? Is it malignant? Well, finding that answer, that kicks off a whole investigation. And just like any good detective story, it all starts with collecting the clues. And this right here, this is the golden rule for this entire process. You have to think of the pathologist as the forensic expert. They're usually miles away in a lab, and they have never, ever seen your pet. So the quality of their diagnosis, it depends completely on the quality of the information they get from the detective on the scene. And that's your veterinarian. Okay, so the first step isn't what you might think. It's not a needle, it's not a scalpel, nope. It's actually more like a pen and paper. It's all about building a really comprehensive case file on our number one suspect, the lump itself. Now, if you look at what goes into this case file, you start to see how every little detail matters. I mean, species and breed? That's critical. Some dogs, like Bernese Mountain Dogs, for instance, are just predisposed to certain kinds of cancer. Prior history is huge too, especially medications. Get this, corticosteroids can actually cause the cells in a lymphoma to break down, literally hiding the evidence from the pathologist. And even something as simple as a little diagram or a photo showing exactly where that lump is, that can be the crucial clue that helps crack the whole case. And this slide just illustrates that point perfectly. Look, on its own, a suspicious-looking cell under a microscope is, well, it's just a mystery. But take that exact same cell and add a case file that says from an older Bernese Mountain Dog, whoa, suddenly that tells a completely different story. The pathologist's suspicion immediately narrows, and the path to a real diagnosis gets a whole lot clearer. All right, the case file is built. Now it's time for the detective on the scene, the vet, to get their hands dirty. The next step is all about collecting the physical evidence. We're talking about the sample itself. You know, this is a much bigger challenge than you might think. A tumor isn't just some uniform blob. Oh no, it's often a really messy, mixed bag of different zones. Some parts are just packed with clues, while other parts are, well, they're biological dead ends. So where on earth do you take the sample from? So it all comes down to strategy. The vet has to be like a skilled marksman, aiming for those good zones, the parts that are rich with living tissue and actual tumor cells. At the same time, they've got to avoid the bad zones, like areas of dead tissue or places with a ton of inflammation or bleeding. Taking a sample from one of those spots is like trying to analyze a corrupted computer file. You just get a lot of noise and no clear answer. Now, there are basically three main ways to collect this evidence. You've got cytology, which is the quickest and least invasive, just a quick poke with a fine needle. Then there's a biopsy, which takes a small core of tissue and gives the pathologist a lot more information to work with. And finally, you have an excision, where the entire mass is removed. That gives you the most complete picture possible. Okay, so you've got the perfect sample from the absolute perfect spot. Fantastic. But if you mess up this next step, it was all for nothing. The evidence has to be preserved perfectly for its journey to the lab. And here it is, the single most important rule of evidence preservation in pathology. The ratio of formalin, that's the preservative, to the tissue has to be at least 10 to 1. Seriously, 10 parts formalin for every 1 part of tissue. If there isn't enough, the tissue starts to break down, to self-destruct, and all those tiny cellular details the pathologist needs to see, they're lost forever. And preservation doesn't happen in a flash. That tissue needs to sit in its formalin bath for at least 24 hours. This gives the preservative enough time to soak all the way through the sample, hardening it and locking all the cells in place. It's like perfectly preserving the scene of the crime for analysis. And here's a funny little inside tip from the lab. Vets are always warned. Avoid jars with narrow necks, like old jam jars. Why? Well, a soft, squishy piece of tissue can be squeezed in pretty easily. But after 24 hours in formaldehyde, that same tissue becomes firm and rigid. And guess what? It's impossible to get it out without literally breaking the jar, which, as you can imagine, is a total nightmare and a safety hazard in the lab. So the sample has arrived at the lab, perfectly preserved. Now we get to go behind the scenes inside the forensic lab and see how the pathologist pieces all these clues together to finally identify our culprit.
But before we go any deeper, we have to talk about this really critical concept, surgical margins. When a vet removes a tumor, they also take a little border of what looks like healthy tissue all the way around it. The pathologist's big job is to check that border, that edge, to see if any cancer cells made it that far. Clean margins. That's the verdict every single pet owner is hoping to hear.
But wait, how does the pathologist even know which edge is which once it's in a jar? Well, vets have some really clever techniques. They might paint the different edges of the mass with different colors of a special India ink that won't wash off. Or if there's one specific spot they're really worried about, maybe where it felt closest to the muscle, they'll put a tiny stitch, a suture, right on that spot. It's like putting a little evidence flag on the sample that screams, "Hey, look right here."
Now, to really understand why those little markers are so important, look at this number. 5 micrometers. That's how incredibly thin the slice of tissue is that the pathologist puts under the microscope. It's unbelievably thin. So they can't possibly slice up and check every single cell on the surface of that tumor. It's just not possible. They have to rely on the vet's markings to guide them to the most critical areas to investigate. Sometimes the cells just look so weird and jumbled up, it's really hard to tell where they even came from. That's where this advanced forensic testing comes in. Pathologists use these special antibodies that act like molecular dyes, and they only stain very specific types of cells. For example, cytokeratin will light up epithelial cells, helping to confirm it's a carcinoma. Vimentin can help identify sarcomas. And then there's Ki-67, which is really cool. It stains cells that are actively dividing, which helps the pathologist figure out just how aggressive the tumor is. All right, all the evidence has been collected, it's been preserved, and it's been analyzed. The detective work is officially done. Now it's time for the final report, the verdict that's going to determine what happens next for the pet.
So the key is to translate this lab speak into something we can all understand. Clean margins is the absolute best news. It means it looks like the whole tumor was removed. Marginal surgery? Well, that means tumor cells were found right at the very edge, and another surgery might be needed. And vascular invasion? That's a big red flag. It means cancer cells were spotted inside blood or lymph vessels, which raises the risk that the cancer could spread or metastasize. And this might be the most important takeaway of all. A pathology report isn't like a blood test that spits out a simple number. It's a highly trained expert's interpretation of visual evidence. You could have 2 amazing pathologists look at the same complex slide and have slightly different interpretations. It's absolutely a science, but it's one with a real element of professional judgment and, yes, subjectivity.
But here's the good news. The tools for this investigation are getting more and more powerful every year. The future is all about personalized medicine. We're heading into an era where we can use advanced genetic sequencing on these tumors, not just to get a more precise diagnosis, but to actually predict how a tumor is going to behave and to pick the perfect treatment for that specific animal. Which really just leaves us with one final thought. As these amazing new tools like AI and genetic sequencing become more common, how is that going to change the job of the pathology detective? It's a really exciting future for veterinary medicine, and one that promises even better outcomes for the pets that we all love.